Okay, so we can start the session. I'll admit people as and when they come in. Can anybody please unmute and let me know if my screen is visible and if I'm audible? Anybody?
Yes, ma'am. Video screen visible and audible.
Thank you, thank you. So we'll start today's session. Good evening, everyone. My name is Aishi Dasgupta, and I'm a PMRF NPTEL TA, PMRF Research Scholar at the Department of Chemistry, IIT Bombay. I'm also an NPTEL TA for this course, and we'll start another live session today where we'll be discussing some of the sample problems. Hopefully, this will help you solve this year's current assignments as well. So let's move to the first question that we have for today. One second. Yeah. So viruses always require a host to replicate. Is it true or false? What do you think? Anybody? Anybody? It's really easy. Just let me know what you think, and I'll let you know what I think. Do you think viruses can be called living beings on their own, or how do they work? What is the basic principle of a viral infection? Can they survive on their own without infecting any kind of cell, be it bacterial or human, mammalian, anything? Bacterial or eukaryotic, basically. Do you think viruses can survive on their own, or do they always require someone else to replicate?
It is true.
Yeah, it's true. Yeah, so viruses always require a host to replicate. They cannot be termed as living beings as such because they cannot survive on their own. So this is a general schematic of how a virus invades a cell. Obviously, this varies from virus to virus and all the hosts that they can infect. But in general, virus is an infectious microbe consisting of a segment of nucleic acid, either DNA or RNA, maybe single-stranded or double-stranded, and is generally surrounded by a protein coat. And it cannot replicate alone. Instead, it must infect some cell and host cell and use components of the host cell, basically hijack the host cell machinery to make copies of itself. And often a virus ends up killing the host cell in the process, causing damage to the host organism. So well-known viruses causing human diseases can be AIDS virus, which is the HIV virus, human immunodeficiency virus, COVID-19 caused by SARS-CoV-2, measles, smallpox. So all this started when in 1898, Frederick Loeffler and Paul Eros found evidence that the cause of foot and mouth disease in livestock was an infectious particle, but it was way smaller than a bacteria. So this was the first clue to the nature of viruses, genetic entities that lie somewhere in the gray area between living and non-living states, because it cannot be completely called a living entity because it cannot survive on its own. So viruses depend on the host cells that they infect to reproduce, but when found outside of the host cell, it acts as a protein coat or capsid, sometimes enclosed within a membrane, enveloped or non-enveloped viruses. We know there can be these two types of viruses based on the envelope coating, and the capsid encloses either DNA or RNA, which codes for the virus elements. While in this form outside the cell, the virus is basically metabolically inert. So that is why it requires a host cell. And so we know that viruses infect living cells. They're incapable of surviving independently and require a host to conduct their life processes. Also, they are kind of parasites. Don't you think they are parasitic in nature and require the host to provide energy and replicate? Basically, they hijack the host cell machinery and produce molecules for their own benefit, for the viral reproduction required for viral process and reproduction. And when they are independently there, they are considered non-living and reproduce only by infecting host cells. So generally, when it comes into contact with a host cell, it can insert its genetic material into the host, literally taking over the host functions, and the infected cell will produce much more viral protein and genetic material instead of its usual products. And some viruses may remain dormant outside host cells for long periods, causing no obvious change in their host cells, which is called a lysogenic phase. But when a dormant phase is stimulated, it enters the lytic phase, and new viruses are formed, self-assemble, and burst out of the host cell, killing the cell and going on to infect the other neighboring cells. So that is how basically a bacteriophage works. Bacteriophage is a virus only, but it infects bacteria, and it roughly measures 200 nanometer. And as we know, viruses can infect both bacteria and like eukaryotes. Bacterial viruses are called bacteriophages, but in eukaryotes also it causes a number of diseases in humans like smallpox, common cold, chickenpox, influenza, herpes, AIDS, example, and even some types of cancer, like the Epstein-Barr virus. It basically leads to a cancer. Even some types of cancer, not all, some types of cancer are linked to viruses. So it's very fascinating. You can go and read online. And viruses themselves have no fossil record, but it is quite possible that they have left traces in the history of life. And it has been hypothesized that viruses may be responsible for some of the extinctions seen in the fossil record. It was once thought by some that outbreaks of viral disease might have been responsible for mass extinctions, such as the extinction of the dinosaurs and other life forms. It's still a hypothesis, and this theory is basically hard to test, but seems unlikely since a given virus can typically cause disease only in one species or in a group of related species. Even a hypothetical virus that could infect and kill all dinosaurs 65 million years ago could not have infected the ammonites or foraminifera that also went extinct at the almost the same time. So it seems unlikely, but it's still a theory. On the other hand, because viruses can also transfer genetic material between different species or host, they are extensively used in genetic engineering as well. So humans have found out a way to actually, like make use of a virus also. So since viruses can inject the genetic material into the host, so this has been this has been used to our advantage by the scientists. So viruses can actually work out as natural genetic engineering products. A virus may incorporate some genetic material from its host as it's replicating and transfer this genetic material to a new host, even a host completely unrelated to the previous host. And this is known as transduction. And in some cases, it may serve as means of evolutionary change. Although it's not clear how important evolutionary mechanism transition actually is, but viruses are being used extensively by scientists for their own benefit now. So apart from being a really infectious particle, it's also a very huge it has also been a huge breakthrough in genetic engineering. So this is general diagram of virus life cycle. Basically it attaches virus attaches to the host cell wall. For example, if I'm talking with respect to COVID-19, the SARS-CoV-2 virus, it has a spike protein which attaches to a very specific receptor on the cell, ACE2 receptor. But there have been studies where it's shown that even without the ACE2 receptor, the virus can actually interact with the host cell. Then there's a penetration happening when the nucleic acid genetic information moves through the cell membrane into the host cell. Then there's replication, also called biosynthesis. The virus forces the host cell machinery to take over and replicate the viral products instead of its usual products. Then there's the assembly phase or the maturation phase, when newly produced virus components are assembled into new viruses. And then there's a lytic phase, or the release of viruses happen from this host, and these viruses then go off and infect a neighboring cell. So this is a general diagram of virus life cycle. Obviously some things may vary depending on the virus and the host, but this is a general schematic. Okay, moving on to the next question. Viruses are counted by PFU and bacteria are counted by CFU. This is true. PFU is plaque forming unit and CFU is colony forming unit, for information. So do you think it's true? Has anybody worked with viruses and bacteria in labs? Then it will be easy for you to answer. Anybody? Sorry?
True.
Yeah, true. So PFU, or a plaque forming unit, is a measure used in virology to describe the number of virus particles capable of forming plaques per unit volume. And it is basically a proxy measurement rather than a measurement of the absolute quantity of particles, basically, because it's kind of relative measurement. So viral particles that are defective or which fail to infect their target cell will not produce a plaque and thus will not be counted. So it's basically an indirect method of counting. For example, a solution of suppose Lick-borne encephalitis virus with a concentration of 1000 PFU per microliter indicates that 1 microliter of the solution contains enough viral particles to produce 1000 infectious plaques in a cell monolayer. but no inference can be made about the relationship of plaque forming unit to number of virus particles. The concept of plaque forming unit uh, of virus is equivalent to the concept of colony forming units of bacteria. So this is how the dilution occurs. It's quite the same for CFU only. The plaque forming unit has another step basically. So if this is a virus stock, then you are basically diluting the virus stock, and then you are mixing the virus dilution with the cells, whichever cells it infects. If it infects amoebal cells, then you are plating with amoebal cells. If it infects bacterial cells, you are doing the same with bacteria. If it infects mammalian cells, you must know the host of the virus, and you're plating it with these, and then you're overlaying the cells with agarose. Then you're removing the agarose layer, and then you're staining the cells to visualize plaques in the monolayer. And virus titer is determined by counting the number of plaques and multiplying by the dilution factor. Plaque counts from at least 3 replicates at each dilution should be averaged. Sorry, I'll just 1 second. Yeah. So plaque countings from at least 3 dilutions should be taken, because one dilution might be any kind of error from your side as well. There might be pipetting error or anything, but when you're averaging from 3 dilutions, it's most likely what is happening. You're basically taking an average. The chances of error are going down. So this is how plaque forming assay is done. Obviously, it will vary from virus to virus, and there will be established protocols in the internet. So if you're particularly working on a particular virus, then you can actually go through literature review and find out if people have done that before. And this is the colony forming unit, pretty much the same, just that in here you basically you take the sample, whatever it is. For example, let's take a drinking water, a water sample, and you're diluting the water sample with the autoclaved water. Autoclaved water is not supposed to have any kind of bacteria or any kind of saline. We have done with autoclaved water. You can do it with saline also. And then after diluting, you're pouring it in Luria agar plates, and then you're checking the number of colonies, and you're doing various dilutions, and you're calculating calculating by multiplying the dilution factor or something, and then you can actually get the number of colonies. So this is how it happens. So this is what is known as a colony forming unit. And there's another term which is quite important, which is called the MOI, or the multiplicity of infection, which is a term that describes the number of virions that are added to the cell during infection experiment. So if you are adding any kind of bacteria to a cell that gets infected by the bacteria, or a virus to a cell that gets infected by the virus, we have this term called MOI. For example, if 1 million virions are added to 1 million cells, then the ratio is 1, because 1 million infectious particles are added to 1 million host cells. Ratio is 1:1. And the formula, if you are infecting any kind of bacteria with a virus, the formula for MOI can be PFU by CFU. PFU is the plaque forming units, measure of the number of infectious viral particles. CFU is the colony forming unit. It's a measure of the number of viable cells, bacteria, fungal, mammalian, etc. And there's another procedure called Farge titering, which is a procedure used to quantify the density of plaque forming units, analogous to bacterial cultures colony forming units in a given lysate. So to achieve this, what the researcher performs the experiment, he exposes the exponentially growing bacteria to the bacteriophage of interest before allowing the bacteriophages to propagate overnight. So this is how it happens. And the quantification of viruses involves the number of virus particles in a sample, as I said, and it is an essential methodology used in any virology lab. And in principle, two methods are generally used for the quantitation of virus particles or virus infectivity. There can be molecular methods such as PCR or RT-PCR. There can be virological methods such as plaque assay, which I already talked about, or endpoint dilution assay, or hemagglutination assay. And although the molecular methods have largely replaced the classical virological methods because of the accuracy, they still cannot measure the infectivity or defining feature of the virus particle. So I already talked about plaque forming assay. Like typically tenfold dilution, serial dilutions of the virus stock are inoculated into each plate, and the virus infected cell will lyse and spread the infection to the adjacent cells. And as the infection to lysis cycle is repeated, the infected cell area will form a plaque which can be seen visually. And the PFU per ml result represents the number of infectious particles in the sample, assuming there's an assumption that each plaque is caused by a single infectious virus particle. That's an assumption. Like as we say that each colony is formed from one single bacteria. So that's how it happens. And note that plaque assay is restricted to the viruses that induce cell lysis or death, such as P. coronavirus, influenza virus, herpes virus. So only when cell lysis or death occurs due to the infection of the virus, that that's the only time you can do a plaque assay. What if the virus, like bacteriophage, what if the bacteriophage prefers the lysogenic cycle? So in that case, it will get incorporated as instead as a prophage. It will not lyse the cells. How will you see that using a plaque acid? It will not lyse the cells, so basically no plaque will be formed, but there is viral infection. What if it is staying in the lysogenic phase? So at that point of time, molecular methods like RT-PCR and these things are very are acceptable, because in RT-PCR you will know the particular sequence that you are trying to amplify, and if that particular sequence gets amplified, then that means that particular viral sequence is present inside the cells, and that is present because infection has taken place. So in that case, molecular methods are acceptable. So that's why in today's time, majorly the molecular methods have replaced the cultural traditional classical methods, but still these methods are still quite in use in most of the labs all around the world. Okay, so moving on to the next question. Biomagnification is a prerequisite for bioremediation. What do you think? Anybody? I'll talk about what is biomagnification, what its effects are. Okay, so it's if we talk about strictly being a prerequisite, then the option will not be correct. But we'll first need to talk about what is biomagnification. So here's a difference between biomagnification and bioaccumulation. So biomagnification is basically a process of build-up of certain chemical substances or toxins at higher trophic levels of a food chain, and this is also referred to as biological magnification, hence biomagnification. And the chemical substances include certain toxins, heavy metals, mercury, and other harmful products at a higher concentration. And as these substances increase and accumulate, it moves up in the food chain. And when these contaminated substances are consumed by different levels of organisms in a food chain, it results in severe health hazards. On the other hand, what is bioaccumulation? Bioaccumulation is the process of accumulating toxic chemicals such as pollutants, pesticides, and other toxins directly into the human body, either through the air, water, food intake, or directly through the skin. And as this toxic compound accumulates within the human body, it increases the risk of chronic poisoning and other severe health disorders. And this is a schematic talking about the differences. So bioactivation basically occurs in the food chain, like how one particular organism eats, incorporates some toxic chemical within itself. That particular organism is being eaten by another organism higher on the food chain, and thus the biomagnification is happening. And bioaccumulation on the other side is accumulation of toxic chemical within one particular organism itself. Biomagnification is an increase in the level as one moves higher in the food chain, obviously because you're getting more and more of the toxic chemical. In bioaccumulation, it's increasing the concentration of the substance. It's a particular organism in a study, and when the concentration of the toxic chemical increases in that particular organism, that's when bioaccumulation is taking place. In biomagnification, an increase in the concentration of the pollutants as they move from one trophic level to the other is seen, while in bioaccumulation, it's an increase in the concentration of a pollutant in a particular organism only. Food chain. So biomagnification takes place between two given trophic levels, while as bioaccumulation takes place or occurs within a given trophic level. And the example, we can check the example: the transfer of pollutants and other absorbed toxins from the microscopic aquatic organisms into the small fish, which are later consumed by the larger fish and other marine aquatic animals. However, bioaccumulation talks about the buildup of mercury in fishes and other aquatic animals. So there is no concept of food chain over here. So how can pollutants have long-term effects on organisms? So even when the pollutants are not exactly dangerous, that if an organism takes the pollutants in, it will kill the organism instantly. Even if that's not happening, their presence is having a very long-lasting impact on the food chain through this process. So toxins may increase in concentration as they pass up the food chain. So pollutants such as polychlorinated biphenyls or PCBs enter the ocean as industrial waste, and they are absorbed by microscopic phytoplankton at the bottom of the food chain. And even though phytoplanktons absorb only a tiny amount, small creatures called zooplankton eat large quantities of phytoplankton. So you're realizing one phytoplankton will absorb only a tiny amount of the toxic chemical. It will not kill the phytoplankton. But when zooplanktons are then feeding on many small phytoplanktons, they eat a lot of large quantities of the phytoplankton, taking in all the PCBs from what all the phytoplanktons have been eating. And finally, small fish, they feed onto the zooplankton, continuing to magnify the amount of PCBs or polychlorinated biphenyls in the food chain. And in the waters of the Pacific Northwest, apex predators like the killer whale, they end up using like taking in a higher concentration of toxins due to biomagnification. So this is how biomagnification occurs. Whereas in bioaccumulation, they're talking about only one organism in a single organism's body over time. Mercury, for example, is a pollutant that has entered waterways and lakes through industrial processes, and fish and shellfish absorb the mercury directly from their environment. And although they may only absorb small amounts at a time, the mercury can remain in the fish's body for months or even longer, making the effect long lasting. And this leads to the mercury buildup or accumulating in the fish's body, posing a great danger to any organism that eats the fish. Okay. So this is the difference between biomagnification and bioaccumulation. So biomagnification is defined as the rise or increase in the contaminated substances caused by the intoxicated environment. And the contaminants might be heavy metals such as mercury, arsenic, pesticides such as polychlorinated biphenyl, as we talk right now, and DDT. Then these substances are taken up by higher organisms on the food chain, and this is how it gets accumulated. So what are the major causes of biomagnification? Agriculture, obviously. The agricultural pesticides, insecticides, fertilizers, and fungicides are very toxic, and they are released into the soil, rivers, lakes, and seas. And these substances contain small amounts of heavy metals such as mercury, arsenic, copper, lead, cadmium, and these cause several health issues in aquatic organisms and finally humans. Humans feed on fish, right? Marine seafood, and this is how the biomagnification happens. Then, apart from agriculture, there is organic contaminants. So, manures and biosolids are processed industrially and contain contaminants like pharmaceuticals and personal care products. And these substances have a very large adverse impact on the health of humans, animals, and wildlife. Then, there are industrial activities. The industries and factories release toxic substances that are released. One second. The industries and factories release toxic substances that are released into the soil, lakes, and into the soil, lakes, oceans, and rivers. And the gaseous emissions pollute the environment, which enters into the food chain, causing biomagnification. Another factor can be mining activities in the ocean. So, mining activities are carried out in the deep sea to extract metals like zinc, aluminium, cobalt, silver, and gold. And this mining process generates a large amount of selenium and sulfide, which deposits in water and destroys the entire ocean and coastal regions. The level of toxicity is increased. These toxic substances are absorbed by the aquatic organisms higher in the food chain. So these are some of the causes. What are the effects of biomagnification? First is obviously impact on human health. Biomagnification makes humans much more prone to diseases such as cancer, kidney problems, liver failure, birth defects, respiratory disorders, and heart diseases. Effects on reproduction and development of marine creatures. So, the toxic chemicals accumulate in the important organs of aquatic organisms that affect the reproduction and development process. For example, the shells of the eggs of the seabirds are very thin that might get crushed by the birds themselves during incubation, and the toxic chemicals mercury and selenium destroy the reproductive organs of the aquatic creatures. Then another effect is the destruction of the coral reefs. Cyanide that is used in leaching gold and fishing is the main cause of the destruction of coral reefs. Coral reefs are the dwelling and the feeding grounds for many sea creatures, and the destruction affects the lives of many aquatic animals. Then disruption of the food chain. So the chemicals and toxins which are released into the water bodies disrupt the food chain. The small organism absorb the toxins which are eaten up by the larger animals, and these toxins thus get biomagnified into the higher level of organisms. So this is what biomagnification is all about. Bioaccumulation also means an increase in the concentration of the chemical in a single single organism, and compounds accumulate in the living things anytime they are eaten and stored faster than they are broken down. So understanding the dynamic process of bioaccumulation is very important in protecting human beings and any other organism from the adverse effects of chemical exposure, and it has become a clinical consideration in the number of chemicals. So these are some of the terms that are used quite interchangeably, but there's these bioaccumulation and biomagnification differ in a little term. So storage, a term that is sometimes confused with bioaccumulation, means a temporary deposit of a chemical in the body tissue or in an organism. Storage is just one facet of chemical bioaccumulation. Bioconcentration is the specific bioaccumulation process by which the concentration of a chemical in an organism becomes higher than its concentration in the air or in the water around the organism. Although the process is same for both natural and man-made chemicals, the term bioconcentration usually refers to the chemicals foreign to the organism. For fish and other aquatic animals, bioconcentration after uptake through the gills or sometimes the skin is usually the most important bioaccumulation process. We also talked about biomagnification, how a typical food chain includes algae eaten by the water flea, eaten by the minnow, eaten by a trout, and finally consumed by an osprey or a human being. And each step results in increased bioaccumulation, resulting in biomagnification. That the an animal at the top of the food chain, or through its regular diet, may accumulate a much greater concentration of chemicals than present in the organisms lower in the food chain. So this biomagnification process has been illustrated in a study done with DDT, which showed that where soil levels were 10 ppm, 10 parts per million, DDT reached a concentration of 141 ppm in earthworms and 444 ppm in robins. Through biomagnification, the concentration of the chemical in the animal at the top of the food chain may be high enough to cause death or adverse effects or behavior, reproduction, disease, resistance, and this endanger the species throughout. Fortunately, bioaccumulation does not always result in biomagnification. Bioaccumulation begins when a chemical passes from an environment into an organism's cells. And uptake is a complex process which is still not fully understood. And scientists have learned that chemicals tend to move or diffuse passively from a place of higher concentration to one of low concentration. So the force or pressure for diffusion is called the chemical potential. It works to move a chemical from outside to inside an organism. So a number of factors may increase the chemical potential of certain substances. For example, some chemicals do not mix well with water. We know they are called lipophilic, mean fat loving, or hydrophobic, meaning water hating. In either case, they tend to move out of the water and enter the cells of an organism where there's more lipophilic, hydrophobic environments. So the same factors affecting the uptake of a chemical continue to operate inside an organism, hindering a chemical's return to the outer environment. Some chemicals are attracted to certain sites and by binding to proteins or dissolving the fats that temporarily stored. And if uptake is slow or is not continued, or if the chemical is not very tightly bound in the cell, the body can eventually eliminate the chemical. So this is how bioaccumulation occurs. Another factor that affects bioaccumulation is whether an organism can break down or excrete a chemical, which is the term is called metabolism. Basically, this ability varies among different organisms and species and also depends on the characteristics of the chemical itself. Chemicals that dissolve readily in fat but not in water tend to be more slowly eliminated by the body and thus have a greater potential to accumulate. Many metabolic reactions actually change a chemical into more water soluble forms called metabolites, and that are readily excreted. There are exceptions, however. Natural pyrethroids, insecticides that are derived from the chrysanthemum plant, are highly fat soluble pesticides, but they are easily degraded and do not accumulate. The insecticide chlorpyrifos, it is less fat soluble but more poorly degraded, tends to bioaccumulate. So these are the factors affecting metabolism, and this often determine uh, whether a chemical achieves its bioaccumulation potential in a given organism or not. So this bioaccumulation is actually a state of dynamic equilibrium, because when a chemical enters the cells of an organism, it is distributed and then excreted and stored or metabolized. Excretion, storage, and metabolism decrease the concentration of the chemical inside the organism, increasing the potential of the chemical in outer environment to move into the organism. So during constant environmental exposure to the chemical, the amount of chemical accumulated inside the organism and the amount leaving, these reach a state of dynamic equilibrium. So to understand this concept, a very clever analogy was described by one of the scientists. So imagine a tub filling with water from a faucet at the top and draining out through a pipe of smaller size at the bottom. So when the water level in the tub is low, little pressure is exerted on the outflow at the bottom of the tub. As the water level rises, the pressure on the outflow increases. Eventually, the amount of the water flowing out will equal the amount of the water flowing in, and the level of the tub will not change. If the input or the outflow is changed, the water in the tub adjusts to a different level. It is the same concept with the living organisms. An environmental chemical will at first move into an organism very rapidly, then it is stored, degraded, or excreted. With constant exposure, its concentration inside the organism will gradually increase. Eventually, the concentration of the chemical will reach and inside the organism will reach an equilibrium with the concentration of the chemical outside the organism, and the amount of chemical entering the organism will be the same as the amount leaving. Although the amount inside the organism remain constant, the chemical continues to be taken up, stored, degraded, and excreted. And if the environmental concentration of the chemical changes, the amount inside the organism will also change until it reaches a new equilibrium. So exposure to large amounts of chemical for a long period of time may overwhelm the equilibrium, potentially causing very harmful effects. Likewise, if the concentration in the environment decreases, the amount inside the organism will also decline. Should the organism move to a clean environment so that exposure ceases, then the chemical eventually will be eliminated from the body. So these are some of the bioaccumulation factors, and obviously it varies between individual organisms as well as different species. Large, fat, long-lived individuals or species with low rates of metabolism or excretion of a chemical will bioaccumulate more than small, thin, and short-lived organisms. Thus, an old lake trout may accumulate much more than a young bluegill in the same lake. But what is bioremediation now? So bioremediation is a biotechnical process which abates or cleans up the environment, the contamination, not environment, the contamination. Sorry, it's a type of waste management technique which involves the use of organisms to remove or utilize the pollutants from a polluted area. So there are several remedies where contaminated water or solid is purified by chemical treatment, incineration, and then burial in a landfill. So there are other types of waste management techniques which include solid waste management, nuclear waste management, etc. And bioremediation is different as it uses no toxic chemicals. So microorganisms like bacteria and fungi are the main role player when it comes to executing the process of bioremediation. Bacteria are the most crucial microbes in this process as they break down the waste into nutrients and organic matter. And even though this is an efficient process of waste management, but bioremediation cannot destroy 100% contamination. Bacteria can easily digest contaminants like chlorinated pesticides or clean oil spills, but microorganisms fail to destroy heavy metals like lead or cadmium. So this is the process of bioremediation. There are some types of bioremediation such as biostimulation. So biostimulation happens when the bacteria is stimulated to initiate the process. So the contaminated soil is first mixed with special nutrient substances including other vital components either in the form of liquid or gas, and it stimulates the growth of microbes, thus resulting in the efficient and quick removal of contaminants by microbes or other bacteria. This is called biostimulation. Then we have bioaugmentation. In bioaugmentation, at times there are certain sites where microorganisms are required to extract the contaminants. For example, municipal wastewater. In those special cases, the process of bioaugmentation is used. There's only one major drawback in this process. It almost becomes impossible to control the growth of microorganisms in the process of removing the particular contaminant. So in bioaugmentation, you actually put the specific microbe into the polluted area. Then we have intrinsic bioremediation. So this is most effective in the soil and water because of these two biomes which always have a high probability of being full of contaminants and toxins. So the process of intrinsic bioremediation is mostly used in underground places like underground petroleum tanks. So in such places what happens, it is difficult to detect a leakage and contaminants and toxins can find their way to enter through these leaks and contaminate the petrol. There's only microorganisms can remove the toxins and clean the tanks. So bioremediation is a very important branch of biotechnology which uses living organisms basically to decontaminate affected areas in the removal of contaminants, pollutants, toxins from soil, water, and other environments. It's also used to clean up oil spills or contaminated groundwater. So it may be done in situ, that is at the site of contamination, or it may be done ex situ, that is away from the site. So this is how bioremediation occurs. There are various advantages to bioremediation. I'll talk about one particular example. So in 1989, the Exxon Valdez oil tanker ran aground of the coast of Alaska, and the tanker ended up spilling approximately 11 million gallons of oil. Around the same time, bioremediation was gaining traction as a viable option for oil cleanups. So the EPA and the Exxon Mobil Corporation both began testing different compounds. Initial tests regarding the effectiveness of the bioremediation look promising. This is very interesting. You can actually go online and check out what happens in bioremediation. And obviously it's not, the question was, is it biomagnification is a prerequisite for bioremediation? Not necessarily. That's why this is false, but we have now talked a lot about these two terms, so I'll move on to the next question. So the sulfate ion reduces the pH, and the low pH helps in the dissolution of the heavy metals, thereby leading to acid mine drainage. So I'm sorry, I've already shown the answer. What do you think, it's true or false? If you know, then just let me know.
True.
Yeah, so sulfate ion reduces the pH, and low pH will give rise to it will basically give rise to the heavy metals dissolution, and that will lead to acid mine drainage. So this picture is not that clear, but this is what happens in acid mine drainage. It is one of the acid mine is one of the main pollutants causing environmental impact, and its main sources include leachate from the abandoned mines and underground deposits, leach water from open pits, tailing pits, etc., and they have been contaminating the entire environment because of the dissolution of the heavy metals. So this is a very prominent thing that has been actually contaminating the entire environment since a long time. So that is what happens when acid mine drainage occurs. So the SO2, the sulfate, is very important factor regarding that, in place of that, because it's basically one of the primary contributors to acid mine drainage. So that's how it works. So we'll move on to the next question. One way to treat acid mine drainage is to add a reducing layer to reduce the sulfates and prevent reduction in the pH.
True.
Okay, anybody else has any other opinion? Okay, so it is true, and this thing has been in use since quite a long time. So as I said, acid mine drainage is an important environmental concern, and remediation through biological sulfate reduction and metal recovery can be applied for this. Also, there's a microbial community composition has a major impact on the performance of bioreactors to treat AMD. And acidophilic sulfate reducing bacteria are strongly influenced by proton sulfide and organic acids concentration. So this entire phenomena occurs due to the oxidation of metallic sulfide ores, which produce sulfate-rich waters with low pH and high heavy metals content, generally termed as this acid mine drainage. And this is of great environment concern as some heavy metals are highly toxic. So within a number of possibilities, biological treatment applying sulfate-reducing bacteria is an attractive option to treat AMD or acid mine drainage and to recover metals. So the process produces alkalinity, neutralizing the AMD simultaneously, and the sulfide that is produced reacts with the metal in solution and precipitates them as metal sulfides. So this phenomenon is characterized by low pH, higher concentration of sulfate and heavy metals. It's very widespread. So there are sulfate reducing passive bioreactors that have received much attention lately as promising biotechnologies for AMD treatment, and they offer advantages such as high metal removal at low pH, stable sludge, very low operation cost, and minimal energy consumption. And sulfide precipitation is a desired mechanism of contaminant removal. However, many mechanisms, including adsorption and precipitation of metal carbonates and hydroxides, occur in passive bioreactors. And the efficiency of sulfate reducing passive bioreactors is sometimes limited because they rely on the activity of an anaerobic microflora, including the sulfate reducing bacteria, which is controlled primarily by the reactive mixture composition. And the most important mixture component is the organic carbon source. The performance of the field bioreactors can also be limited by the acid mine drainage load and metal toxicity. And several studies conducted, actually they have found out that the best mixture of natural organic substrates for sulfate reducing bacteria are still in question, and there are many critical parameters for this design and long-term operation. And there obviously needs to be additional work done to properly assess the long-term efficiency of reactive mixtures and the metal removal mechanisms. So this is how it occurs. The pollution occurs inevitably during the extraction of mineral resources, and that's why acid mine drainage is a very challenging factor that has come up on the global mining industry. That occurs due to the oxidation of the sulfide minerals when exposed to air, water, and microbial activity, and it's widespread in active or even abundant polymetallic sulfide and coal mines. Its strongly acidic pH value is commonly below 4, so you can see it's very strongly acidic, and it's a generally high concentration of sulfate and dissolved metal ions. However, the discharge of untreated acid mine drainage has the risk of contaminating nearby water sources, flora and fauna. And this has negative impact not only on the biodiversity but also causes high levels of metal ions in the soil to reach the top of the food chain, again by biomagnification. Ultimately, it will create a risk to the human health as well. So it's one of the very important challenges that has been taken over by that occurs due to this, and so it's one of the main pollutants causing environmental impact, and its main sources I've already talked about. So how to treat AMD? So AMD is acid mine drainage. So water flowing through the active or the abundant mine sites can become polluted by the material being mined. So the sulfuric acid is produced when water interacts with sulfur bearing materials in the presence of oxygen and common bacteria. So the acidic water now formed by this process can dissolve many metals in bioavailable forms, posing a threat to humans and wildlife. So, and if acid mine drainage gets into the surface waterways, both the acidity and the metal content can produce significant problems. So there are some approaches that are being taken to prevent and to clean up the acid mine drainage, and most of these try to focus on trying to neutralize the acid, prevent exposure of mine materials to oxygen and flowing or percolating water basically, or otherwise prevent bacteria from catalyzing the necessary reactions. So some common methods include reclamation of the contaminated land by adding lime or any other alkaline materials to neutralize the acidity, and adding uncontaminated topsoil, planting vegetation and modifying slopes to stabilize the soil basically and reduce infiltration of the surface water into the underlying contaminated water. Or there can be soil removal to relocate contaminated material to new sites where it can be monitored and treated. Then there can be direct treatment of the contaminated water either through treatment plants where lime or any other neutralizing materials are added, like to reduce the acidity which causes metals to precipitate out of the contaminated water, or there can be artificial wetlands which will help sequester contaminated material in place and where microbial action produces oxygen free conditions to help prevent further sulfuric acid formation. Then you can fill in abundant mines and materials that will prevent the formation of acid mine drainage. So this can include flooding the mines with water to remove the oxygen necessary to form acid mine drainage, or filling in mines with alkaline materials to prevent the formation of acidic water. Or else you can do relocation and isolation of mine waste that may produce acid mine drainage if allowed to react with water. So this often involves isolating the waste from interaction with groundwater by moving the waste above the water table, treating it and covering it with a layer of impermeable material to keep out surface water. Then there's bacterial control. So certain common bacteria substantially speed up the formation of acid mine drainage. Some efforts to prevent acid mine drainage involve the use of bactericides to kill these special bacteria, or the addition of organic waste to provide an alternative energy source for some of these bacteria and produce oxygen free conditions that prevent the formation of sulfuric acid. And obviously another option can be diverting the water from the mine site to prevent it from running through AMD forming materials, or disposing mine waste underwater to prevent exposure to oxygen. In addition to all these methods that I discussed right now, a wide variety of alternative methods are also being employed to prevent these. So the preventive measures will generally focus to prevent the production of acid effluents from the mine. On the other hand, corrective measures will be designed to treat the acidic waters produced by mining so that they stop being a threat to the environment. And it's easy to understand that when planning the mine abandonment, priority should be given to suitable preventive techniques. So the use of corrective measures is only justified for the most serious and unforeseen causes of acid water pollution. Therefore, the mine waste treatment must be included in the mining project in order to minimize the exposure of these materials to inclement the weather, and also to reduce the mine waste volume to be processed and stored. So I've already talked about how AMD treatment can cause a significant on the economic performance also, and preventing techniques can act on any three elements essentially in the formation of acidic waters, oxygen, water, or any kind of bacteria that help in this. So there can be there's a technique called surface water diversion. So in this technique they basically try to reduce the water supply to those potentially acid generating materials by using safeguarding channels, pipes and waterproof channels, slope changes around the material area, etc. So the application of this technique should be preceded by accurate hydrological and hydrogeological studies, and it is precisely the lack of these which sometimes causes unsuccessful application of this method. And the systematic application of this technique can be is prone to errors basically, because an effort which will also cost a lot of money to divert the surface water to reduce large masses of water supply to areas where subsequent acid production is not extremely high can lead to the reduction of the quantities of water entering areas that are priorly are most dangerous, as they can have an extremely high production of acid. So even so this is a cheap technique, easy to apply, and that works well in conjunction with other techniques, it still has its own problems. Then we have soil compaction. So in soil compaction, the compaction of the mining waste decreases their permeability. So this compaction achieves on the one hand the reduction of water flow, which remains longer in the tailings, hindering the oxidation of the materials and therefore the rate of acid production, and on the other hand the decrease of oxygen diffusion, which slows down pyrite oxidation and makes the tailings less habitable for ferrooxidant bacteria. So the decrease of the porosity means a decrease of both hydraulic conductivity and oxygen diffusion. So the oxygen diffusion decreases by 3 to 4 orders of magnitude when the with the degree of saturation. That water content reaches 85% or so. So although this is not itself a restorative method, it works effectively with any other techniques that we have in use. Sorry, one second. Then another technique is the technique of dry covers, which this question talks about adding a reducing layer, or basically the application of dry covers can be done with the aim of different objectives. One, mining waste stabilization to prevent erosion by wind and water. Two, because of an aesthetic appearance improvement. Three, both prevention and inhibition of pollutant release. So this can be achieved with methods that prevent waste penetration by oxygen and water. So without oxygen, the chemical and the microbial oxidation of sulfide minerals decreases, and a minimized water flow causes a reduction in the drainage water production, so that the metals are spread very slowly, resulting in a dilution with time. And there are a multitude of cover designs that cover the waste with soil or rock and use the cementitious materials to form a crust. And the effectiveness of this method increases when choosing a high and dry location for the dump. And the cover should be stable, provide a long-term protection, and this generally requires at least two layers of different materials, as it is rare that the required properties are found together in the same material. First, a bottom sealing layer with low permeability is required to decrease the oxygen diffusion and to hold large amounts of water, acting as a barrier to oxygen. Suitable materials for this layer are clays, especially bentonite. And however, these clay barriers are particularly prone to breaking during dry conditions, letting in oxygen and water. So organic materials may be a good substitute to clay, provided that the layer is thick enough. And covers rich in organic materials have been used to act as a radon barrier. Some artificial materials such as plastic membranes have higher impenetrability than natural materials. However, care must be taken to prevent membranes from physical damage. So there are also doubts about how long it will last before the chemical and the microbial degradation appears. So above the low permeability layer, a drainage layer can be placed to protect it from destruction by erosion, drying, freezing, and influence of man-made errors, animals and plants, and microbial destruction. So this drainage layer can also act to prevent the upward capillary migration of metals and salts. Additionally to this physical layer, there are chemical covers where minerals are added to form an impenetrable crust, and furthermore additional measures can be taken to increase the reduction in the flow of oxygen, as the addition of oxygen consuming microorganisms or the compaction of the mining waste. So this is how covers can be done. There is another process where the covers with sludge happens. So sludge occurs as a cover insulating material, and it has been studied since 1989. And in this study, the mud used to cover the residue from a treatment plant in a nearby factory was stabilized by the addition of fly ash produced by the combustion of coal and wood from factories. So as fly ash contains calcium oxide, it can therefore increase the pH. Furthermore, a protective layer composed of tree bark waste overlaps on the layers of the sludge. So these are some of the techniques. They can also seal with clay. They are handling tailings and everything. So this is one of the techniques to reduce acid mine drainage. Moving on to the next question. Which one of the following types of reactors relies on biofilms? Anybody? Anybody knows the answer? Anybody? You know what is
Nutrient removal.
okay. Anybody else? So we know what is a biofilm. We have talked about this. So we'll talk about the reactors. So the answer is actually trickling filter, and I'll let you know what a trickling filter is and why is it used in wastewater treatment. So trickling filters are basically also termed as biological trickling filters. They are used to remove organic compounds from wastewater. Basically, it's an aerobic treatment device that removes organic material from wastewater by using microorganisms attached to a medium. So this mechanism is employed in many different technologies, including rotating biological contactors and packed bed reactors. So these processes are known as attached growth processes. Suspended growth processes, on the other hand, are systems that keep microorganisms alive in a solution. So how municipalities use trickling filters? So in many circumstances, settled sewage reaches a high level and flows into the basic settlement tank. And the supernatant from the tank flows into a dosing device, which is often a tipping bucket that distributes flow to the filter's arms. The water flush goes through the arms and exits via a series of downward pointing holes, and this rotates the arms, distributing the liquid uniformly across the surface of the filter material. So most are open to the elements and are easily vented to the atmosphere. So pollutant removal from the wastewater stream involves both absorption and adsorption. of organic molecules as well as certain inorganic species such as nitrite and nitrate ions by the layer of microbial biofilm. So typically the filter material is designed to have a very high surface area to volume ratio. And typical materials are also very porous, have a large interior surface area in addition to the medium's outward surface. The passage of wastewater through the medium supplies dissolved oxygen, which the biofilm layer requires for biochemical oxidation of organic molecules, as well as carbon dioxide gas, water, and other oxidized and products. So as the secondary sludge develops, the biofilm layer sloughs off into the liquid flow and becomes part of it. And to separate and remove the sloughed film, a trickling filter is frequently followed by clarifier or sedimentation tank. So other filters that employ high density media like sand, foam, and peat moss do not produce sludge and must be cleaned, but they do require forced air blowers, backwashing, or enclosed anaerobic environment. So, what are the different types of biological trickling filters? Modest home septic tank outputs and extremely small rural sewage treatment systems can be treated using single trickling filters. And many trickling filters are often used in tandem in large centralized sewage treatment plants. Structure is configured for the single pass operation in which treated water is applied to the trickling filter once before being discharged, or for multi-pass operation in which a part of the treated water is cycled back and retreated via closed loop. And there are also multi-pass systems which help improve the quality and reduce the total nitrogen levels by encouraging nitrification in the aerobic media bed and denitrification in the anaerobic septic tank. So some systems operate the filters in two banks in series, allowing the wastewater to pass through the filter twice with the sedimentation step in between. And to equalize this load, the filters are rotated every few days. Because much of the carbonaceous oxidative material is eliminated on the first pass through the filters, this kind of treatment can enhance nitrification and denitrification. So this is what trickling filter is all about. It's a type of wastewater treatment system. It consists of a fixed bed of rocks, coke, gravel, slag, or polyurethane foams, magnum, peat moss, ceramics, or plastic media sewage or other wastewater flows downward and cause a layer of biological film or a microbial slime biofilm to grow covering the bed of the media. Then aerobic conditions are maintained by splashing diffusion and either by forced air flowing through the bed or natural convection of the air if the filter medium is porous. And the treatment of the sewage or other wastewater with trickling filters is among the oldest and most well developed techniques. And the terms trickling filter or trickling biofilter are all interchangeably used. So I had another image, I don't know why it's not showing. So this is how basically a trickling filter works. And so the removal of pollutants from the wastewater will include both absorption and adsorption of organic compounds and some inorganic species as well. And it happened by the use of microbial film or the biofilm, and the filter media is typically chosen to provide a very high surface to volume ratio. And typical materials are often porous and have considerable internal surface area in addition to the external surface of the medium. And the passage of the wastewater and over the media provides dissolved oxygen, which the biofilm layer later requires for the biochemical oxidation of the organic compounds and releases CO2 gas, water, and other oxidized end products. And as the biofilm layer thickens, it eventually sloughs off into a liquid flow and subsequently forms part of the secondary sludge. So typically a trickling filter is followed by a clarifier or sedimentation tank for the separation and removal of the sloughed film. And filters utilizing the higher density media such as sand, foam, and peat moss do not produce a sludge that must be removed, but many require forced air blowers, backwashing, and enclosed anaerobic environment. So the biofilm that develops in a trickling filter may become several millimeters thick and is typically a gelatinous material that may contain many species of bacteria, ciliates, and even protozoa, amoeboid protozoa, annelids, roundworms, insect larvae, other microfauna. So this is very different from many other biofilms which may be less than 1 millimeter thick. Within the biofilm, both aerobic and anaerobic zones can exist, supporting both oxidative and reductive biological process. And at certain times of the year, especially in the spring, rapid growth of organisms in the film may cause the film to be too thick and it may slough off in patches, leading to the spring slough. So this is what biofilm usage in the trickling filter happens. Moving on to the next question, match and pair. Grid chamber, aeration tank, primary sedimentation tank, and nitrate. Which ones do you think match properly?
Option 3.
Option 3, which is 1, 4, 2, 1, 3, 2, nitrate. Yes. So you're correct. So this is how the match and pair happens. So we know that sewage treatment or domestic wastewater treatment, municipal wastewater treatment is basically a type of waste treatment which aims to remove contaminants from sewage to produce an effluent that is suitable to be discharged to the surrounding environment, or an intended reuse application, thereby preventing water pollution from raw sewage discharges. And this sewage that contains wastewater from different households, then businesses, and possibly various industrial wastewater. So there are a number of sewage treatment processes to choose from. These can range from decentralized systems, like including on-site treatment systems, to large centralized systems involving a network of pipes and pump systems which convey the sewage to a treatment plant. And for cities that have a combined sewer, the sewers will also carry urban runoff to the sewage treatment plant. And sewage treatment often involves two main stages called the primary and the secondary treatment, while advanced treatment also incorporates a tertiary treatment stage with polishing process and nutrient removal basically. So secondary treatment, what it does, it can reduce the organic matter from sewage using aerobic or anaerobic biological process. That's what the secondary treatment does. And a number of sewage treatment technologies have been developed, mostly using biological treatment methods. And design engineers and decision makers need to take into account technical and economical criteria of each alternative. So often the main criteria of selections are desired effluent quality, expected concentration, operating costs, availability of land, energy requirements, sustainability aspects. So in many developing countries and rural areas with low population densities, sewage is often treated by various on-site sanitation systems and not conveyed in sewers. So these systems include septic tanks connected to drain sites, on-site sewage systems, vermifier systems, etc. At the global level, I think an estimate of 52% of sewage is treated. However, sewage treatment rates are highly unequal for different countries. So what does happen? It basically takes involves two main steps: the primary and the secondary treatment. And as I said, advanced treatment will also incorporate a tertiary stage. So in the preliminary stage, which is called the pre-treatment, it removes, where is it? It is called pre-treatment. This is the grit chamber. It removes coarse materials that can, be easily collected from the raw sewage before they damage or clog the pumps and sewage lines of primary treatment clarifiers. So the influent is a sewage water that passes through a bar screen to remove all the large objects like cans, rags, sticks, plastic bag, etc., carried in the sewage stream. And this is most commonly done with an automated mechanically racked bar screen in modern plants serving large populations, while in smaller or less modern plants a manually clean screen may be used. So the raking action of the mechanical bar screen typically plays according to the accumulation on the bar screens and flow rate, and the solids are collected and later disposed in a landfill or incinerated. And bar screens or mesh screens of varying bar screens or mesh screens of varying sizes may be used to optimize the solid removal. And if gross solids are not removed, they become entrained in the pipes, and this makes moving parts of the treatment plant and can cause substantial damage and inefficiency in the process. So what is grit removal? There was a question, grit chamber. One of the options was grit chamber, which has matched with sand and silt. So what happens in the grit chamber? So the grit consists of sand, gravel, rocks and other heavy materials. So this preliminary treatment may include a sand or grit removal channel or chamber where the velocity of the incoming sewage is reduced to allow the settlement of grit. And grit removal is necessary to reduce formation of deposits in the primary sedimentation tanks, or reduce the frequency of tank cleaning caused by excessive accumulation of grit, and or to protect moving mechanical equipment from abrasion and accompanying abnormal wear. So the removal of grit is essential for equipment with closely machined metal surfaces such as conveniators, fine screens, centrifuges, heat exchangers, and high pressure diaphragm pumps. Grit chambers thus come in three types. It can be horizontal, vertical, horizontal vortex, or aerated grit chambers. So that is the number. Then happens flow equalization. So equalization basins can be used to achieve flow equalization, and this is especially useful for combined sewer systems which produce peak dry weather flows or peak wet weather flows. In some larger plants, there are fat and grease are also removed by passing the sewage through a small tank where skimmers collect the fat floating on the surface, and air blowers in the base of the tank may be used to help recover the fat as a froth. Many plants will also use primary clarifiers with mechanical surfaces skimmers for fat and grease removal. Then comes the primary treatment. What is that? Primary treatment. So primary treatment is the removal of a portion of the suspended particles and organic matter from the sewage. So it consists of allowing sewage to pass slowly through a basin where heavy solids can settle to the bottom, while oil, grease, and lighter solids float to the surface and are skimmed off. And these bases are called primary sedimentation tanks or primary clarifiers. You can see how the heavy things have sedimented in the bottom, and this is what is happening. They typically have a hydraulic retention time of 1.5 to 2.5 hours, and the settled and the floating materials are removed, and the remaining liquid may be discharged or subjected to a secondary treatment. And primary settling tanks are usually equipped with mechanically driven scrapers that continually drive the collected sludge towards a hopper in the base of the tank, basically in the base of the tank, to remove where it is pumped to sludge treatment facilities. So the base of the tank, and it is further pumped to sludge treatment facilities. Now, sewage treatment plants that are connected to a combined sewer system sometimes have a bypass arrangement after the primary treatment unit. This means that during very heavy rainfall events, the secondary and the tertiary treatment systems can be bypassed to protect them from hydraulic overloading, and the mixture of the sewage and stormwater receives primary treatment only. So the primary sedimentation tanks remove about 50 to 70% of the suspended solids and 25 to 40% of the biological oxygen demand. Then we move to the secondary treatment. So the main process involved secondary treatment are designed to remove as much of the solid material as possible. So they use biological process to digest and remove the remaining soluble material, especially the organic fraction. And this can be done with either suspended growth or biofilm process. The microorganisms that feed on the organic matter present in the sewage grow and multiply, constituting the biological solids. So these grow and group together in the form of flocks or biofilms, and same specific process granules. The biological flock or biofilm and the remaining fine solids from a sludge can be finally separated and settled. And after separation, liquid remains that is almost free of solids and with a greatly reduced concentration of pollutants. So this secondary treatment can reduce organic matter from sewage using aerobic or anaerobic processes, and the organisms involved in this process are sensitive to the presence of toxic materials. So then we move to the tertiary treatment, which is present in only very advanced systems, and this is a basic system. There might be some intermediate stages as well. So the purpose of the tertiary treatment is to provide a final treatment stage to further improve the effluent quality before it is discharged into the receiving body or used. So more than one tertiary treatment process may be used at any treatment plant. If any disinfection is practiced, it's always the final process called effluent polishing. Tertiary treatment may include biological nutrient removal, disinfection, removal of micropollutants such as environmental persistent pharmaceutical pollutants, and tertiary treatment is sometimes defined as anything more than secondary and primary treatment in order to allow discharge into the highly sensitive or fragile ecosystems such as estuaries, low flow rivers, or coral reefs, etc. So this is what happens. After that, there is a disinfection stage as well. So disinfection stage is used to kill pathogens prior to disposal, and it is increasingly effective after more elements of what is happening. So here we can see that the first chamber, grid chamber, it removes the sand and silt. We have already seen over here how it removes the grid disposal happens. Then the aeration tank helps in BOD thing, basically the biological oxygen, the number of microorganisms. That is what happens in the BOD tank, aeration tank. It's a part of the secondary treatment. Then there's a primary sedimentation tank which removes suspended impurities. This will remove any kind of suspended impurities by, like, the heavy things will go down and the suspended things will go up, and the middle liquid will actually flow to the next part. And nitrate removal or various nutrient removal happens in the tertiary treatment stage, which is not shown in this particular diagram, but tertiary treatment happens in some kind of advanced plants. So she's correct. The match and pair she said was correct. Moving on to the next question. Select the correct sequence for the treatment process for the preliminary treatment is to be given for sewage. What do you think is the, I just showed you the schematic, so what do you think is the correct answer? I showed you the schematic just now. Anybody? The answer is screening.
Option 2.
Screening, grit removal, correct. Option 2. Screening, grit removal, primary sedimentation, aeration, secondary sedimentation. That is what is happening. There is a screening, grit removal, primary sedimentation, aeration, and then secondary sedimentation. So the option is what she said is correct. Option 2 is the correct one. So I already showed you the schematic. So this is how the entire thing happens: bar screening, grit removal, primary clarifier, aeration, secondary clarifier, then disinfection, and everything else is happening also. Moving on to the next question: which of the following pairs is correctly matched? Okay, so it will be tertiary treatment, removal of nutrients, because it happens in some kind of advanced systems only, but the tertiary treatments is actually there to actually remove any kind of nutrients that are not really there for the purpose. So the tertiary treatment basically provides a final treatment stage to further improve the effluent quality before it is discharged, and more than one tertiary treatment process can be used at any treatment plant. So if disinfection is practiced, it's always the final process, but that is how tertiary treatment happens. And the biological nutrient removal also happens with the help of tertiary treatment, and so that is why the correct answer will be tertiary treatment. So you can see this is another schematic that I found on the internet based on the sewage treatment plants. Moving on to the next question: are the following statements true? Anybody? First of all, what is BOD and COD? So the aquatic organisms depend on the oxygen present in the water, which is termed as dissolved oxygen content, for the respiratory needs basically. So the amount of dissolved oxygen in a water body depends on the water temperature, the quantity of sediment, the amount of oxygen taken out from the system, and the amount of oxygen put back into the system. The respiration and decaying of organisms take oxygen out of the system, and photosynthetic organisms, aeration, stream flow, return oxygen back to water. So bacteria decompose natural organic debris or detritus as well as organic waste in water by using this dissolved oxygen. So BOD and COD are two measurements which describe the demand of dissolved oxygen by bacteria in the water. So BOD refers to the biological oxygen demand, and COD is the chemical oxygen demand. And the main difference between BOD and COD is that BOD is the amount of oxygen which is consumed by the bacteria while decomposing organic matter under aerobic conditions, whereas COD or chemical oxygen demand is the amount of oxygen required for the chemical oxidation of total organic matter in water. So these are the differences between BOD and COD. So based on that, anybody has any knowledge of this question? So answer is true. Both of the statements are true. So this is a schematic that talks about the difference between BOD and COD. You already talked about it. So COD is the amount of oxygen required for the oxidation of total organic matter, whereas BOD is the oxygen that will be consumed by the bacteria while decomposing organic matter under aerobic conditions. So from the name also it's self-explanatory that one is biological oxidation process, while is chemical oxidation process. And there are chemical test methods have names. And the BOD is determined by incubating a sealed water under specific temperature sample for 5 days and measuring the loss of oxygen from the beginning of the test. While COD is determined by incubating a closed water sample with a strong oxidant like potassium dichromate in combination with boiling sulfuric acid under a specific temperature for a specified period of time. BOD requires 5 days for determination. COD requires the measurement to be taken from a few days. The permissible limit for BOD is 30 mg per liter, whereas for COD it's 250 to 500 ppm. And obviously the BOD value is way lower than the COD value. That's the opposite. For COD, it's always greater than the BOD value. The BOD is capable of oxidizing natural organic detritus and the organic waste in water, whereas COD is capable of degrading industrial sewage but does not measure the oxygen consumption by acetate. So what is the interrelationship between BOD, COD and TOC, total oxygen content? So the typical values for the ratio of BOD and COD for untreated municipal wastewater are in the range of zero point 0.3 to zero point 0.8. So if the BOD to COD ratio for untreated wastewater is 0.5 or greater, the water is considered to be easily treatable by biological means. If the ratio is below 0.3, either the waste may have some toxic components or acclimated microorganisms that may be required for stabilization. The corresponding BOD total oxygen content ratio for untreated wastewater varies from 1.2 to 2.0, and in using these ratios it is important to remember that they will change significantly with the degree of treatment that the waste is undergone. So you see, BOD and COD measurements are taken to determine the pollution level, and like aerobic water purification will always incline more towards the BOD, because BOD is about the biochemical oxygen demand, and this is the type of wastewater and how the BOD to COD ratio or the BOD to TOC ratio will occur. So this is what happens. So this is statement. First statement is true. For the sludge floccules, the sludge floccules are actually, they behave as transporters for aerobic bacteria oxidizing the organic matter. So they have the aerobic bacteria which will actually oxidize the organic matter and actually will clean the treatment plant. So what happens is that aerobic water purification is based on biological oxidation with active sludge, and it is primarily used to remove organic fraction from wastewater. And in addition to the removal of organic substances, this purification method is also able to eliminate nitrogen and phosphorus to a lesser extent, though to a lesser extent. So sludge floccules contain aerobic microorganisms, and this active sludge is a mix of microscopic organisms such as bacteria, protozoa, rotifers, etc. And if sufficient oxygen is present, these organisms are able to oxidize organic compounds in the wastewater into CO2 and water, and this process referred to as dissimilation. However, part of the organic material is used to create new biomass, and this is referred to as assimilation. The rising sludge concentration can be controlled by regularly removing the biomass from the system. However, if an aerobic purification system is only operated at a very low load, there will often be low sludge growth as well. So in the activated sludge process, microorganisms are mixed with wastewater, and the microorganisms come in contact with the biodegradable materials in the wastewater and consume them as food. In addition, the bacteria develop a sticky layer of slime around the cell wall that enables them to clump together to form biosolids or sludge that is then separated from the liquid phase. The successful removal of waste from the water depends on how efficiently the bacteria can consume the organic material and on the ability of the bacteria to stick together, form floc, and settle out of the bulk fluid. So the flocculation or the clumping characteristics of the microorganisms in activated sludge enable them to amass to form solid masses large enough to settle to the bottom of the settling basin. As the flocculation characteristics of the sludge improves, so is the improved settling and improved wastewater treatment. And after the aeration basin, the mixture of microorganisms and wastewater, that is mixed liquor, flows into a settling basin or clarifier where the sludge is allowed to settle. And some of the sludge volume is continuously recirculated from the clarifier as a returned activated sludge back to the aeration basin to ensure adequate amounts of microorganisms are maintained in the aeration tank. The microorganisms are again mixed with incoming wastewater where they are reactivated to consume organic nutrients. Then the process starts again. So the activated sludge process under proper conditions is very efficient. It removes almost 85 to 95% of the solids and reduces the biochemical oxygen demand about the same amount. And the efficiency of this system depends on many factors, including wastewater climate and characteristics. Toxic waste that enter the treatment system can also disrupt the biological activity. Waste heavy in soaps or detergents can cause excessive frothing and thereby create aesthetic or problems. In areas where industrial and sanitary waste are combined, industrial wastewater must often be pre-treated to remove the toxic chemical compounds before it is discharged into the activated sludge treatment process. Nevertheless, microbiological treatment of wastewater is by far the most natural and effective process of removing waste from water. So this is why both these statements are true. Moving on to the next question. In order to remove which of the following nutrients is the anaerobic tank placed prior to the anoxic aerobic tank? Anybody? Take your time and think if you know the answer.
Phosphorus.
Okay, anybody else? Yeah. So phosphorus is the correct answer. So there's a process called EBPR, which is enhanced biological phosphorus removal. It's basically a sewage treatment configuration applied to activated sludge systems for the removal of phosphate. So the common element in this EBPR implementation is the presence of the anaerobic tank where nitrogen and oxygen are absent prior to the aeration tank. And under these conditions, a group of heterotrophic bacteria called polyphosphate accumulating organisms or PAO are selectively enriched in the bacterial community within the activated sludge. And in the subsequent aerobic phase, these bacteria can accumulate large quantities of polyphosphate within their cells, and the removal of phosphorus is said to be enhanced. So generally speaking, all bacteria contain a fraction of phosphorus in their biomass due to its presence in cellular components such as membrane phospholipids and DNA. So therefore, as bacteria in a wastewater treatment plant consume nutrients in the wastewater, they grow and phosphorus is incorporated into the bacterial biomass. So when this PAO or polyphosphate accumulating organisms grow, they not only consume phosphorus for cellular components but also accumulate large quantities of polyphosphate within their cells. Thus, the phosphorus fraction of the phosphorus accumulating biomass is 5 to 7%. And in mixed bacterial cultures, the phosphorus content will be maximal, like 3 to 4% of the total organic mass. And if additional chemical precipitation takes place, for example, to reach discharge limits, the P content could be higher, that is not affected by ABPR. And this biomass is then separated from the treated water at the end of the process, and the phosphorus is thus removed. Thus, if PAOs are selectively, enriched by this EBPR method, considerably more phosphorus is removed compared to the relatively poor phosphorus removal in conventional activated sludge systems. So this is called anaerobic anoxic aerobic method. So in this method, it's one of the various activated sludge methods in the wastewater treatment process. This method is a wastewater treatment method whose main purpose is to remove nitrogen and phosphorus from wastewater, and a method improved from the conventional activated sludge process. This is one of the most advanced wastewater treatment method which removes nitrogen and phosphorus. Red tide in sea is caused by eutrophication, that nutrient salts such as phosphorus and nitrogen flow into the sea. So in this wastewater treatment method, as I said, main purpose is to remove nitrogen and phosphorus, and the reaction tank is configured by 3 tanks which are called anaerobic tank, anoxic tank and aerobic tank. Since no air is sent into the anaerobic tanks and there is no oxygen, microorganism and the activated sludge becomes less active and release phosphorus. Now sewage in anaerobic tanks is sent to the anoxic tank after releasing phosphorus, then sent to the aerobic tank, and agitators are settled in anaerobic tanks, and activated sludge in the tanks are kept being agitated. Now, similar to the anaerobic tank, anoxic tank is a tank with no air sent in and no oxygen water in the tank. And since microorganisms which prefer anaerobic state consume oxygen, nitrate, iron, and nitrate containing liquid pumped from aerobic tank is decomposed in the anoxic tank. And during this decomposed process, nitrogen is released to air and nitrogen gas by being removed its oxygen with nitrate ion. The inside of the aerobic tank is in the state that aeration is done and the oxygen are added into the water. And activated sludge and microorganisms which is active in the aerobic tank decompose ammoniacal nitrogen sent from anoxic tank and change into the nitrate ion. And nitrate containing liquid with the nitrate ion is circulated to anoxic tank with pumps. Also in aerobic tank, activated sludge and microorganisms which have taken in the phosphorus are sent to final settling tank and sedimentate. Now clarified water in the final settling tank is discharged after passing through disinfection apparatus, and some of the settled activated sludge is returned to the adduction tank as return sludge. And then remains sludge sent to sludge thickening tank as waste sludge. So sludge thickened in the thickening tank are dehydrated and treated as dehydrated sludge as it is being dried. So this process of removing nitrogen is also called recirculating denitrification system, since nitrification and denitrification is nitrate containing liquid is circulated. So in the case that only nitrogen is aimed to be removed, reaction tank is configured with only anoxic tank and aerobic tank, and anaerobic tank is not used. But here anaerobic tank is also used because phosphorus removal is also taking place. So this is how the entire anaerobic, anoxic, aerobic method works out. Good. So moving on to the next question. Dissimilatory sulfate reducing bacteria are dash microorganisms and use sulfate as a terminal electron acceptor. Anybody?
Anaerobic.
Okay. So the answer is anaerobic, correct. So I'll explain a little bit. So as we know, the sulfate is present in foods, beverages and drinking water, and its reduction and concentration in the gut depend on the intestinal microbiome activity, especially sulfate reducing bacteria, which can be involved in inflammatory bowel disease as well. Now, assimilatory sulfate reduction is present in all the living organisms, and in this process, the sulfate is reduced to hydrogen sulfide and then included in cysteine and methionine biosynthesis. However, in contrast to the assimilatory sulfate reduction, the dissimilatory process is typical for sulfate reducing bacteria. A terminal product of this metabolism pathway is hydrogen sulfide, which can be involved in gut inflammation and also causes problems in industry due to the corrosion effects. So there are many reviews online you can go and search where they have actually compared assimilatory and dissimilatory sulfate reduction, and this process occurred in some species of the intestinal bacteria such as Escherichia, Desulfovibrio genera, these genera, and the main attention in those reviews are focused on the description of the genes and their location in the selected strains and how it happens. So in dissimilatory sulfate reduction, only a special group of organisms can do this, can extract energy from dissimilatory sulfate reduction, while the assimilatory sulfate reduction process abundant among other organisms, even plants and bacteria. So sulfate reducing bacteria are an important group of microorganisms in various ecosystems, including the intestinal tract, because they require inorganic sulfate as an electron acceptor to obtain energy from organic compounds. And sulfate reducing bacteria are a significant part of the environment because they require inorganic sulfate for energy production. So this process that includes several reactions providing energy for the creation of adenosine triphosphate in the bacterial cells of sulfate reducing bacteria. So sulfate is used as a terminal acceptor and provides energy for growth, and is released from the oxidation of organic compounds such as lactate, propionate, butyrate, or hydrogen. And reduction of the sulfate occurs over many different intermediates that are not released to the environment. So the process of dissimilatory sulfate reduction includes the transfer of 8 electrons. And enzymes that are associated with this process are localized in the cytoplasm or the periplasm of the bacterial cells. And the first step is sulfate and proton absorption into the bacterial cells. After this process, it can be divided into 6 stages, as we can see from the schematic over here. And dissimilatory sulfate reduction is basically a form of anaerobic respiration, as she said over here. It's basically anaerobic because it uses sulfate as a terminal electron acceptor to produce hydrogen sulfide, and this metabolism is found in some types of bacterian archaea, which are often termed as sulfate reducing organisms. The term dissimilatory is used when hydrogen sulfide is produced in an anaerobic respiration process. By contrast, the term assimilatory is used in relation to the biosynthesis of organosulfur compounds. Even the hydrogen sulfide may be an intermediate. It's also an intermediate, but basically organosulfur compounds, the cysteines, are formed. And the dissimilatory thing can happen in 4 steps: that the conversion of the activation of the sulfate to adenosine 5' phosphate or the adenosine 5' phosphosulfate via sulfate adenylyl transferase. Then reduction of this APS happens to sulfite via adenylyl sulfide via adenylyl sulfate reductase. Then transfer of the sulfur from to the sulfur atom of the sulfide to the DSRC protein, creating a trisulfide intermediate catalyzed by DSRAB. And then finally reduction of the trisulfide to sulfide and reduced DSRC via membrane bound enzyme, which is another complicated name, MKJOP. So this is what happens in this thing, and the protein complexes responsible for these chemical conversions are found in all currently known organisms that perform dissimilatory sulfate reduction. And energetically, sulfate is a poor electron acceptor for microorganisms, as a sulfide redox couple is, I think, minus 506 millivolts, which is too negative to allow reduction by NADH or ferredoxin. They are the primary intercellular electron mediators, and to overcome this issue, sulfate is first converted into APS by the enzyme called ATP. Enzyme is called ATP sulfurylase or SAT, at the cost of a single ATP molecule, as you can see. And the APS sulfide redox couple has a minus 60 millivolts thing, so which allows APS to be further reduced by either NADH or reduced ferredoxin using the enzyme adenylyl sulfate reductase, which is APR, which requires the input of 2 electrons, as you can see from here. And the final step, sulfide is reduced by the dissimilatory sulfide reductase or DSR to form sulfide, requiring the input of 6 electrons. This will have 6 electrons. Anyway, this is basically an anaerobic respiration process, so that's why the answer will be anaerobic. She's correct. Moving on to the next question. Which of the following statements is true? Statement A or statement B? Anybody? Anybody? Which one do you think is true? Even if you think only one is true, just let me know. Okay, so both these statements are independently true, but they have no relation to each other whatsoever. So, a simple activity such as disinfecting water in the swimming pool can result in the formation of disinfection byproducts. Very true, because disinfection byproducts are some things that can damage the environment, human health, and trigger antibiotic resistance. Also, the entire genome has been sequenced. Is also true. So the disinfection byproducts are basically formed when disinfectants like chlorine interact with natural organic materials in water, such as in chlorinated drinking water and chlorine-treated swimming pools. Okay. So disinfection byproducts can be found in the air during activities such as showering, bathing, dishwashing, and swimming. And the most common type of disinfection byproducts are the trihalomethanes. How are people exposed to these disinfection byproducts? So they are exposed to by drinking chlorinated water or brominated water, and by breathing in air containing this disinfectant byproducts. The skin also absorbs these byproducts during bathing and swimming, and after exposure this remain in the body for only a short period of time. But how do these DPBs or disinfection byproducts affect people's health? So chronic exposure to these DPBs can actually even result in the increase the risk for certain types of cancer. And humans exposed to initially large amounts of some DPBs or disinfection byproducts could experience liver damage and decreased nervous system activity. So there was in the US population there was a study in the fourth national report on human exposure to the environmental chemicals. The CDC scientists measured 4 different trihalomethanes in the blood of at least 1,200 participants, who were aged from 20 to 60 years, who took part in the National Health Nutrition Examination Survey. I think during the year 2003 or 2004. And by measuring the trihalomethanes in blood, scientists were able to estimate the amounts of trihalomethanes that have entered people's bodies. So, CDC scientists did not find detectable blood levels of trihalomethanes in all participants. Levels were similar to those seen in the small past studies. Now, finding a measurable amount of one or more trihalomethanes in blood does not imply that the levels of disinfection byproducts cause an adverse health effect. Now, biomonitoring studies of blood trihalomethanes can provide physicians and public health officials with reference values so that they can determine whether people have been exposed to higher levels of trihalomethanes or not than found in normal general population. So biomonitoring data can also help scientists plan and conduct research work on exposure and health effects. So these are some of the disinfection methods and the chemicals used for these disinfections. So chlorination method is a very common and very frequently used methods for disinfecting swimming pools. So chlorination method, that's why people are asked to wear shower caps during swimming, because it damages your hair. Even shower caps don't help as much because your hair will get wet eventually. I have done swimming myself, and shower caps do not help that much. It doesn't prevent your hair from getting wet completely, but at least it's better than going without a shower cap. That will completely damage your hair for a person who is swimming regularly. So the chlorination methods for the disinfection of swimming pool water can lead to the formation of potentially toxic disinfection byproducts such as trihalomethanes I've already talked about, haloacetic acids, haloacetonitrile, then trihaloacetaldehyde and chloramines. And with the awareness that the demand of alternatives to chlorine disinfection has already increased, and the alternative methods aimed at improving the quality of swimming pool water are electrochemically generated mixed oxidants, ultraviolet irradiation, UV based advanced disinfection, oxygen versus ozone, UV based AOPs, etc. And the use of ozone and UV radiation for the disinfection of swimming pool water has been adopted in some cases, though together with chlorine for the provision of a residual disinfectant. However, chlorination methods for disinfection are widely still used in practice. And the efficiency of the ozonation process can be increased by combining ozone with hydrogen peroxide and UV such as O3H2O2 or O2UV, and although substantial research has been done on the applications of these things, for even ozone etched hydrogen peroxide and ozone UV process for the disinfection of drinking water as well, a limited number of studies is available on the disinfection of swimming pool water. So, like, I think Glauner and Christensen, they have studied a lot. There's a paper, Chima et al. They have also studied this. So similarly, significant research has also been done on the use of UV raised AOPs for the disinfection of drinking water. But in swimming pool water, there is lesser number of studies. Moreover, the studies conducted on the use of these things for the treatment of swimming pool water only investigated few disinfection byproducts such as trihalomethanes and haloacidonitriles. So it is recognized that the conditions for the treatment of drinking water are completely different from the treatment of swimming pool water. So compared with drinking water, swimming pool water disinfection byproducts have their own distinct characteristics due to the different nature of organic precursors and continuous loading of dissolved organic carbon and dissolved organic nitrogen, which are released by swimmers. So all these factors add an additional complication to the disinfection and toxicological safety of the swimming pool water. For instance, the reported level of total oxygen and total organic carbon (TOC) in tap water and swimming pool water was in the range of zero point 0.3 to 1.4 and zero point 0.5 to 7 mg per liter respectively. Similarly, the level of nitrate nitrogen compared it was higher in swimming pool water compared to tap water. Consistently, the reported level of total nitrogen in drinking water was lower compared to the swimming pool water. The temperature of swimming pool water is generally higher, 25 to 35 degrees Celsius, compared with drinking water, which is generally from 1 to 23 degrees Celsius, which is an important parameter to restrain the continuous anthropogenic pollutant release and increases demand for higher doses of disinfectants due to the higher rates for decay, for instance, in the case of chlorination. So the reported level of free residual chlorine in the tap water and swimming pool water can be in the range of 0.03 to 0.57 and 0.24 to 1.4 mg per liter respectively. So the pH of tap water is generally 6.8 to 7.8. It's lower than the swimming pool water pH, which is 7.6 to 8.2. So studies of drinking water disinfection cannot be exactly replicated to optimize the treatment conditions of swimming pool water. And in the last few years, research has been increased on alternative methods for the disinfection of swimming pool water. There are several publications available online. You can actually go and check out how these things work. Although the comparative analysis of different disinfection methods within studies is available, it has still not been done between the studies. For example, a comparison between chlorination, ozone chlorination and some UV radiation, these have all not been performed as such. So a comprehensive and critical review of the performance of all of these is required, and that's how the people will get to know about the differences. But still, chlorination, as I said before, only it's the most commonly used method of disinfection for the prevention of waterborne diseases and inactivation of pathogenic microorganisms in swimming pools. However, the use of chlorination has also many drawbacks, such as presence of chlorine-resistant microorganisms such as Cryptosporidium pavum and Giardia lamblia. These are all chlorine-resistant, and the formation of potentially toxic byproducts. So, like, past research has potentially identified, toxic. Almost 100 byproducts toxic in pool water samples, and among the known carbonaceous byproducts, the most common are the trihalomethanes and also haloacidonitriles. So these are some of the drawbacks of chlorination. So the first option is true. The second statement is the entire human genome has been sequenced. So we all know about the Human Genome Project. It's a massive discovery in the field of science. So getting the entire human genome sequenced is very fascinating. It's a great achievement in the field of science. It's amazing. Like, even centuries later, people will talk about this advancement. So the Human Genome Project is one of the greatest scientific feats in history, and the project was a basically voyage of biological discoveries led by an international group of researchers looking to comprehensively study all the DNA of a select set of organisms. So it was launched in October, late 1900s, I think October 1990, and it was completed in April 2003. And the Human Genome Project's signature accomplishment was generating the first sequence of the human genome. It provided fundamental information about the human blueprint, which has since accelerated the study of human biology and improved the practice of medicine. You have no clue how much it has improved. And the Human Genome Project was completed in 2003. You can see this is a basic timeline. I urge all of you to go back and check these landmarks. It's very fascinating. But the completion happened in 2003 April, but still it covered 92% of the total human genome sequence. And the technologies to decipher the gaps that remained didn't exist at that time. But scientists knew that the last 8% likely contain information important for fundamental biological process. And since then, there's been a lot of development, scientific development. Researchers have developed better laboratory tools, computational techniques, strategic approaches, and the final complete human genome sequence was described in a set of 6 papers in April 1st, 2022, just last year. It's not even a full year exactly, and they were described in a paper in a set of 6 papers, all published in Science, definitely, and companion papers were also published in several other journals. The work was done by the Telomere to Telomere Consortium, and T2T, it's called T2T. T2T is led by researchers at the NIH, National Human Genome Research Institute, the University of California, Santa Cruz, and the University of Washington, Seattle. NHGRI was the primary funder. The short read technologies were originally used to sequence the human genome. These provide several hundred bases of DNA sequence at a time, which are then stitched together by computers. Such methods still leave some gaps in genome sequences. So, over the past 2 decades, two new DNA sequencing technologies emerged that can read longer sequences without compromising accuracy. The PacBio Hi-Fi DNA sequencing method can read about 20,000 letters with nearly perfect accuracy, and the Oxford Nanopore DNA sequencing method can read even more, up to 1 million DNA letters at a time with modest accuracy. So, both of these were used to generate the complete human genome sequence, and in total the new project added nearly 200 million letters of the genome genetic code. So this last 8% that still remained from 2003 of the genome includes numerous genes as well as repetitive DNA sequences which may influence how cells function, and most of the newly added sequences were in the centromeres, the dense middle sections of chromosomes, and near the repetitive ends of each chromosome, that's the telomeres. So the complete genome sequence will be particularly valuable for studies that aim to understand how DNA differs from person to person. For example, T2T, or the telomere to telomere researchers, use the sequence as a reference to discover more than 2 million previously unknown sequence variants in the human genome. These included variants with many medically relevant genes, and this complete human genome sequence has already provided much newer insight into genome biology. And scientists are actually looking forward to the next decade of discoveries about these newly revealed regions. So it's like one of the consortium co-chair, Dr. Adam Philippi, has said that truly finishing the human genome sequence is like putting on a pair of glasses. Now they can clearly see everything. They are one step closer to understanding what it all means. And this accomplishment can now serve as a model for sequencing genomes from globally diverse people, a goal researchers are pursuing. So further work is also needed to finish the complete sequence of the Y chromosome, which was not contained in the cells used for this study. So this foundational information will strengthen the many ongoing efforts to understand all the functional nuances of the human genome, which in turn will empower genetic studies of human disease. So these are some of the things that this is why I think people do science. It's so fascinating because now they are one step closer to understanding what happens in certain diseases, how the genome can be the genome genetic variants happen in different diseases, and the next decade which will further work on this developments will be increasingly fascinating. So I'll urge all of you to go and check this landmark in genetics and genomics thing, and it's very fascinating. Just read it like a storybook, and it's really interesting. So yes, this sentence, second statement is also true. The entire genome, human genome, has been sequenced. So these are both independently true statements. Moving on to the next question. Which of the following statements is not true? Anybody? Is not true. Anybody try? Anybody? Any option you think is not true?
Option 2.
Option 2 is not true. Okay. Anybody else? We'll talk about all the options and what do these mean. So the correct answer will be option 2. Uh, so what are endocrine disruptors? The first option was regarding endocrine disruptors. So many chemicals, both natural and man-made, may mimic or interfere with the body's hormones, known as the endocrine system. Now, endocrine disruptors are basically chemicals that are linked to developmental, reproductive, brain, immune, and other problems. And endocrine disruptors are found in many everyday products, including some plastic bottles, containers, liners of metal food cans, detergents, flames, retardants, food, toys, cosmetics, and whatnot, even pesticides. So, some endocrine disrupting chemicals are slow to break down in the environment. Their characteristic makes them potentially hazardous over time. And endocrine disrupting chemicals cause adverse effects in animals, but limited scientific information exists on potential health problems in humans. So, because people are typically exposed to multiple endocrine disruptors at the same time, assessing public health effects is difficult. So, what are the common endocrine disruptors? So, BPA. BPA, it's very, you can check out any plastic bottle of yours, just check out the below part, just invert the plastic bottle and check out the below part, you can see the BPA content. So, what is this BPA? BPA is bisphenol A. It's used to make polycarbonate plastics and epoxy resins, which are found in many plastic products, including food storage containers. Then another can be dioxins. Dioxins are used, basically produced as a byproduct in herbicide production and paper bleaching. They're also released into the environment during waste burning and wildfires. Then there is perchlorate, which is a byproduct of aerospace, weapon, and pharmaceutical industries, and they are found in drinking water and fireworks. There is perfluoroalkyl and polyfluoroalkyl substances. These are widely used in industrial applications such as firefighting foams, non-stick pans, paper and textile coatings. Then there are phthalates, which are used to make plastics more flexible, like they are found in food packaging, some kids' toys, medical devices, etc. Cosmetics also. Then there are phytoestrogens. So they are naturally occurring substances in plants that have hormone-like activity, such as genistein and diaziene. I'm not sure about the pronunciation. Diaziene, that are in soy products like tofu or soy milk. Then there are polybrominated diphenyl ethers, which are used to make flame retardants for household products such as furniture, foam, and carpets. Then you have PCB, polychlorinated diphenyls, which are used to make electrical equipment like transformers and in hydraulic fluids, heat transfer fluids, lubricants, and plasticizers. Then we have triclosan. It may be found in some antimicrobial and personal care products like body wash, liquid body wash. So how do people like human beings, how do we get ourselves exposed to such endocrine disrupting chemicals? So we can get exposed to these disruptors through food and beverages consumed, pesticides applied, and cosmetics used. So in essence, our contact with these chemicals may occur through diet, air, skin, and water. Now, even low doses of these endocrine disrupting chemicals are really unsafe, and the body's normal endocrine functioning involves very small changes in the hormonal levels. Yet, we know even these small changes can cause significant developmental and biological defects. This observation leads scientists to think that endocrine disrupting chemicals and their exposure, even at low amounts, can actually ruin the body's sensitive systems and lead to health problems. So you can see here, when absorbed in the body, an endocrine disruptor can decrease or increase normal hormone levels, mimic the body's natural hormones, and alter the natural production of hormones. It's massive. The destruction is massive. So, and there's recently been a concern regarding pharmaceutical products and personal care products also. So these contaminants of emerging concern, including pharmaceuticals and personal care products, are increasingly being detected at low levels in surface water, and there is concern that these compounds may even have an impact on aquatic and marine life. And it's important for EPA to be able to evaluate the potential of impact of these substances on aquatic life and have an approach for determining protective levels for aquatic organisms. These chemicals have features that require additional consideration when applying existing ambient water quality criteria for the protection of aquatic life. And there are many contaminants of concern that are so-called endocrine disruptors. It alter the normal functions of hormones, resulting in a variety of health effects, and can actually alter the normal hormone levels, leading to reproductive effects in aquatic organisms. And evaluating these effects may require testing methodologies not typically available, along with endpoints not previously evaluated in any guideline. And the emerging contaminants may also demonstrate low acute toxicity, but they can cause significant reproductive effects at very low levels of exposure. And in addition, the effects of exposure to aquatic organisms during the early stages of life may not be observed until adulthood, and therefore traditional toxicity test endpoints may not be sufficiently comprehensive for criteria derivation for these chemicals, and the chemicals may also have specific modes of action that may affect only certain types of aquatic animals. So that's why there are many rules and regulations that are developed by the EPA. So that's how the endocrine disruptors work. The next question is exposome. So what is an exposome? So an exposome is basically, it refers to all the environmental exposures, internal and specific, external, that humans experience from conception through to death. So there are articles that examine the importance of both fundamental biological and public health perspectives within the context of the exposome, dealing with novel and well received concepts that include exposotype, issues of life stage, aging, and both short epigenetic and long term evolutionary biological effects. So the problem of scale use of omics technology and that database resources are also discussed in various papers published online. So special focus is also placed on the role of the ultraviolet light exposure in regard to the photolysis of folate and biosynthesis of vitamin D as important molecular mechanisms within human health and biology. So specifically, this includes critical nutrigenomic interactions. High dimensional biology will obviously permit extremely large scale initiatives in the future that will shed new light on the exposome by better characterizing a plethora of new and established biomarkers. However, smaller delimited studies should not be ignored as they can still help define aspects of the genome exposome that remain unclear. And still, overall trends in the field are moving inexorably towards multiplexing, metabolite, and other omic analysis within the large population studies. So there are articles that actually talk about exposome and everything. So the exposomes, as described by Wild, it encompasses life course environmental exposures, including lifestyle factors, from prenatal period onwards. And under the exposome framework, the totality of the environmental exposures comprise inherent features of exposures and they change over time. So the exposome in this view represents, the totality of the exposure from conception to death as a quantity of critical interest in understanding the environmental causes of chronic diseases. And the concept of the exposome is analogous to the concept of the genome and was developed with the goal of quantifying environmental exposures with the same precision and accuracy as the human genome. However, environmental exposures are more variable than individual genomes, and unlike genomes which are composed of the same chemical entity, DNA, environmental exposures, including chemical exposures, social determinants of health, and lifestyle factors, belong to distinct categories that cannot be readily grouped together. And all genes are comprised of DNA and therefore ontologically homogeneous. They could be analyzed using the same methodological procedures and understood using the same conceptual strategies. Other omics approaches in biology, such as proteomics, lipidomics, and metabolomics, all have succeeded for the same reason because they study substances of the same category: proteins, lipids, metabolites. Such bioomics approaches derive additional clarity by being organ specific. For instance, proteomics of liver could not be merged with proteomics of the kidney or metabolomics of the heart. Thus, grouping all environmental factors into one amorphous set would pose difficulties similar to those that arise from simultaneously, studying all biomolecules, proteins, lipids, metabolites, carbohydrates, etc., without first distinguishing between them on the basis of their biological distinct nature or tissue origin. On the larger scale, this would be akin to studying plant and animal kingdoms as a whole, without first categorizing them into phyla, families, genera, and species. Hence, to redeem this promise of the biomics approach, people need to distinguish between different domains and components of the environment determine how they relate to each other. So that is what exposome basically is. And the exposome model of the environment includes three different categories of environmental exposures: internal, specific external, and general external. The internal environment is thought of as the body's internal chemical environment, self-explanatory, in which the exposures are biologically active chemicals within the body and consist of chemicals produced by metabolism, physical activity, gut microflora, inflammation, and oxidative stress. Specific external exposures consist of environmental pollutants, infectious agents, diet, alcohol, tobacco, smoking, etc. Lastly, general external exposures in the exposome include socioeconomic status, psychological influences, built environment, and climate. Although assessing exposure by measuring the internal chemical environment is attractive and bioactivation of a toxicant could be considered to be part of exposure, tissue levels of the metabolites of external and internal chemicals or even the levels of untransformed chemicals reflect not only the extent of exposure but also the rate of tissue deposition, removal, and metabolism, processes that depend upon an individual's genetics. Genetics. Where was I? Lost the flow. Individuals genetics, age, nutritional status, and co-exposure to other pollutants. So, in other words, the internal chemical environment is only indirectly reflective of actual exposure, and assessing the internal chemical environment exclusively may lead to significant exposure, misclassification. Moreover, internal correspondence is difficult to establish for non-chemical exposures, such as socioeconomic status, social networks, or built environments. And in addition, studying changes in oxidative stress, inflammation, and cytokines, etc., confuses effects with causes. So, changes in the production of reactive oxygen species or cytokines are responses to external exposures, not the exposure itself. Yes, we can learn much about the nature of the exposure by studying its effects, but to avoid equivocation between cause and effect, it may be important to distinguish external environmental exposures from their biological consequences and not to blur the distinction between them. And it is equally important to distinguish between direct and indirect interactions between biological process and the environment. And contrary to the common allusions to gene environment interactions, genes and environment may not directly interact. In some cases, the environment can directly affect genes that by causing gene mutations, but in a majority of cases, genes synthesize proteins, which in turn synthesize lipids, carbohydrates, metabolites, and other proteins, products that respond to changes in the environment and convey this information to genes. And even epigenetic changes associated with chromatin remodeling and DNA repair mechanisms are orchestrated by proteins. So each step of these interactions is regulated by different processes and subject to different constraints that cannot be overlooked in assessing how environmental exposures affect a biological process. So that's why it's important not to equate environmental exposure to specific changes in only one class of biomolecule, that is genes, without considering the entire biological response. So, success in mapping the entire human genome has fostered the complementary concept for the exposome. The exposome can be defined as the measure of all the exposures of an individual in a lifetime and how these of exposures relate to health. An individual's exposure begins before birth and includes insults from environmental and occupational sources. Understanding how exposures from our environment, diet, and lifestyle, etc., interact with our own unique characteristics such as genetics, physiology, and epigenetics impact our health is how that exposome, will be articulated. So exposomics is the study of the exposome and relies on the application of the internal and the external exposure assessment methods. Internal exposure relies on fields of study such as genomics, metabolomics, lipidomics, transcriptomics, and proteomics. And commonalities of these fields include use of biomarkers to determine exposure, effect of exposure, disease progression, and susceptibility factors. So, use of technologies that result in large amounts of data and use of data mining techniques to find statistical associations between these exposures, effect of exposures, and other factors such as genetics with disease. So, external exposure assessment relies on measuring environmental stresses. So, common approaches include using direct reading instruments, laboratory-based analysis, and survey instruments. The extent to which internal and external exposure assessment will contribute to an understanding of the exposome is still being debated, as much approach is has certain merits and demerits. And the key factor in describing the exposome is the ability to accurately measure exposures and effects of the exposures. So many of the omics technologies have the potential to further our understanding of disease causation and progression, like metabonomics and adaptomics, that is DNA and protein adapt measurement, have been used in the past to establish exposure disease relationships. So research is needed to determine the utility of the omics technologies in defining the exposome. So, and why, like your question might be, why should we study the exposome? Because one of the promises of the Human Genome Project was that it could revolutionize our understanding of the underlying causes of disease and aid in the development of preventions and cures for more diseases. However, genetics has been found to account for only about 10% of the diseases, and for the remaining causes appear to be from environmental exposure, environmental causes. So, to understand the cause and eventually the prevention of disease, environmental causes need to be studied. But what are the challenges of advancing exposomics? Some challenges that may limit the progress in this field of study are evident. Like, an individual's exposome is highly variable and dynamic throughout their lifetime. The impact of exposures can also vary with the individual's stage life. For example, exposure to the drug thalidomide during specific developmental periods in utero causes malformation of limbs. Exposure to lead in infants and early childhood can lead to cognitive deficiencies. Exposures during early years may also predispose an individual to certain chronic diseases later in life. So NIOSH has a unique opportunity to attribute occupational exposure information to this emerging field. And the impact of environmental occupational exposures can be different for each individual, obviously, because of differences in genetic and other personal factors. Some people will develop a disease while another person with the same or greater exposure will not. and the exposome may help to determine the underlying causes for this difference. Mapping an entire exposome for an individual will be very difficult, if not impossible, because of the complexity of the lifetime of exposure. Specific exposures can be difficult to measure due to the lack of sensitive methods, or not knowing that an exposure has even occurred. Even when the exposure is known, measuring the exposure can be difficult since the indicators of exposure may be transient, such as for most chemicals, which are rapidly excreted and only a short time period frame exists to directly measure them. In other cases, past exposure can be defined using legacy biomarkers. A common example of a legacy biomarker is antibodies produced by exposures to environmental or occupational insults. So, the experience in studying genetic involvement in diseases serves as a model for studying the relationship between exposures and disease. Uh, Currently, genome-wide association studies are performed with the aid of new technologies, which produce cheaper and faster analysis to generate hypotheses about the relationship between genes and diseases. And these studies identify gene pathways associated with diseases. And when an association has been identified, specific hypotheses about the role of specific genes in the disease can be generated and tested. And an approach to the exposome is to use internal biological media and measure multiple endpoints, and the data will be analyzed, to identify associations between health outcomes and biomarkers of exposures, biomarkers of response, or patterns of biomarkers, etc. So one important aspect of the exposome will be adherent to strict ethical principles as the exposome is deciphered. This will paramount to ensure that the rights of individuals are not compromised when they're determining exposures and relationship to their health. So these are some of the characteristics or features of the exposome. So it is very difficult, like the genome of an organism will stay fixed, right, from one point to the other, but the exposome, the entire exposure is happening at, it differs with your age, it differs with your interest, and it's very difficult to map. And as I already talked about, the most of the diseases have only 10% reliant on the genes can be studied to figure out how diseases happen because of mutated genes and stuff like that, and it only is right for 10% of the diseases. What about the other 90%? Most of it are relying on exposures basically. So cancers can be genetic, it can be hereditary, definitely, but some cancers, or like even the same cancers, some of the contribution can come from the exposures also. How you're leading your life, your lifestyle, your diet style, are you smoking or not, what all heavy metals you're exposed to, what are contaminants are exposed to, all of this will make a sense. So and it's also very difficult because it will not be very general, it will be very person to person specific. Someone who's being exposed to lead for a certain amount of time, someone who's exposed to much more lead might not be showing any signs of the disease, but someone who's exposed to even little amount of lead for a short period of time might get that disease. So it's very person to person specific, and that's why this new term called exposome, and it's much more difficult than sequencing the genome and understanding the impact of the genome, because that stays constant but exposome will not. So that's why it's very difficult, and there are a lot of studies in today's time that are being done in this domain. So these are some of the options. The not true statement was the sulfur oxidizing bacteria. So mostly the sulfur reducing bacteria that leads to corrosion, microbiologically influenced corrosion will happen. It will degrade the environment, integrate the materials, usually metal, due to the activity of the microorganisms, and this not only applies to metals but also to non-metallic surfaces and all. So corrosions will generally happen by microbes, and some of the main types of microbes are sulfur reducing bacteria, even though some sulfur oxidizing bacteria are also involved. So this second statement is not exactly wrong. So I was confused because all these statements seem true. Out of these four, second might be the most relevant, but even some sulfur oxidizing bacteria can lead to difficult, corrosion things. So because for sulfate reducing bacteria, they have long been associated with corrosion of metals exposed to the sulfate containing water, and have been the focus of much biocorrosion research because the problem they've posed to the offshore oil industry. So under anaerobic conditions, we already discussed, the sulfate reducing bacteria uses sulfate as a terminal electron acceptor and a layer of biomolecular oxygen biomolecular hydrogen is present on the metal surface due to the reduction of the hydrogen ions present in the water and the oxidation of the metal. So the sulfate reducing bacteria will oxidize the hydrogen via dissimilatory sulfate reduction, producing sulfide as a waste product. We already talked about dissimilatory sulfate reduction, and sulfide will then react with the metal ions present to produce the metal sulfides, which will further accelerate the oxidation of the metal. And it can also form hydrogen sulfide, which is used by sulfur oxidizing bacteria, further contributing to corrosion. So sulfur reducing bacteria might be starting the process, but sulfur oxidizing bacteria also takes part in the process, because they can they can oxidize hydrogen sulfide to form sulfuric acid, which corrodes steel and concrete in wastewater environments. And hydrogen sulfide gas can diffuse above anaerobic wastewater and dissolve into water droplets or metal surfaces above the sewage. And with oxygen present, sulfur oxidizing bacteria such as acidithiobacillus thiooxidants can then metabolize the hydrogen sulfide gas to sulfuric acid, which has a destructive effect on the exposed concrete and steel surfaces. So that's how microbiologically influenced corrosion can actually occur in aqueous environments as well, and it lead to water treatment and sewage plant. It's also very common in other sectors of the economy, from oil and gas production and systems to underground pipelines and chemical processing. So corrosion actually costs the US government over $200 billion per year, and microbial corrosion often goes undetected in many industrial systems, resulting in significant waste and increased operating costs, which can lead to severe economic losses. So yes, this is how microbial corrosion occurs. So today we'll stop here. Anybody has any question based on what we discussed today, or anything, any doubt? Otherwise, we can stop the session and conclude the session, and we can continue from the next session, which is on next Friday. Anybody? So if you don't have any questions, we can conclude the session. Thank you for bearing with me for 2 hours and being so interactive. We will meet again next Friday, same link, same time. Thank you.